So I made this slide a little larger because I know in the PowerPoint is tiny, very small. So I make it a little larger. And this is your capillary network, and this is at the tissue level. So remember when I did my drawing on the whiteboard last week of the arterioles breaking apart into or branching into capillaries, right? And then those are the ones bringing oxygen and nutrients to the tissues, the cells. And then on the other end of this capillary bed is called, we have the small tiny capillaries from the venous end, and then we have the venules that are going to collect all that carbon dioxide and waste, soluble waste, that is going to return through the vena cava, both superior and inferior, into the right atrium. So are you all there? So can you see yourselves there? Okay. All right. So the picture here, it doesn't have the cells, but you need to imagine the cells are here, right? So when it says tissue fluid, it means there's little interstitial fluid that we have in between the cells. So tissue cells are here. Okay, so this is a branch of the aorta. Obviously, the aorta is the major artery that comes out of the left ventricle. It has all the oxygen, all the nutrients, and the aorta then starts to branch, right? It goes up to branch the shoulders, neck, and head, and then it also comes down with that aortic arch, right? And it comes down into the thoracic, abdominal, pelvic region, and they start branching and branching and branching until we can reach every single cell in our body, right? So this one right here in red is obviously a arteriole, which means it's a small artery, right? From chapter 13, you probably know that the arteries are they have very thick muscular layer, right? It's a muscle layer of smooth muscle. And as we go into smaller and smaller arteries, the muscle layer starts to decrease in thickness. So for example, aorta is the highly muscular type of artery. And then when we start getting closer to the capillary bed, that smooth muscle layer starts to decrease. So when you get to an arteriole, the muscle layer is not as thick. But when you get into the capillaries, it has disappeared completely, right? Capillaries are one cell thick, which means it's only endothelium. And endothelium is simple squamous epithelium. So that's to show you how thin those tiny little capillaries are. And it makes sense because we need to have diffusion through these capillaries, right? We have to make sure the things that our tissues need is getting there. So the layer of that to diffuse has to be extremely thin, right? Our arteries cannot do this because they have the muscle layer, they have the serosa on the outside and the endothelium. So that's very thick. When we get to the capillaries, it's just endothelium. So the diffusion goes and reabsorption goes very fast either way. So that's what we have here. And we have that the capillary on this arterial end is red because again, we're taking oxygen and nutrients, right? Now, if you notice, kind of like in the middle here, it turns a little purple because we don't have a clear break between arterial blood and venous blood, right? It is kind of like in the middle, it's kind of like a mixture, right? And then on the other end, we have these capillaries on the venous end, we call it, which are again, one cell thick, it's only simple squamous. And we have venules, which are small veins, right? And then these ones are gonna become major veins. So when you look at the arterial end, you see different arrows here, right? So you see 3 yellow arrows that are going up, meaning from the capillary to the tissues, right? But we also see these 2 other arrows that are going in. So what it means is opposing forces, right? So on this end, we really want to have a net filtration. We really want to have that diffusion of oxygen and nutrients. But this force that you see here in 3 yellow arrows, they have to overcome these 2 opposing forces. So we're going to see what the opposing forces are. So that's what the little arrows mean, okay? So when we come down here, you have, this is a blood vessel. Remember, the capillary is just simple squamous epithelium. You can see this here. These are the simple squamous cells. Red blood cells or white blood cells are not supposed to go through, right? Whatever is going to go through is just oxygen, nutrients. But if you think about it, the nutrients are dissolved in the plasma, in the fluid portion of the blood. So obviously some tiny little tissue fluid is going to go with it, right? Now remember, this tissue fluid should be mostly reabsorbed by the venous end, because if we start accumulating tissue fluid here, that's what causes edema. So when we analyze what are the forces that allow for this diffusion to happen, and what are the opposing forces of that diffusion. So when we come here where it says blood flow, we are coming with the hydrostatic pressure of the capillary. So this is your PC in this formula right here. So PC, we call it hydrostatic pressure, but if you want to call it blood pressure, it's the same thing. They're calling it hydrostatic because hydrostatic means water. And as you know, most of our blood is water. So that's why they're calling it hydrostatic pressure. But you can call it blood pressure as well, right? So do you remember how is the blood pressure when we are leaving the aorta? What's the value that we have when we leave the aorta, which is our systolic blood pressure? That is what?
120.
120, right? And in some textbooks, they say 115 is considered normal these days, but 115, 120, right? So we come out with 120. By the time we reach the arterial and look at the tissue level, right? Your 120 has become 37. So this is what happens to your blood pressure. Your blood pressure starts high, and as we leave the heart, drops, okay? So it starts to drop. It's a gradient that we have from high to low, right? And when we get to the tissues, your blood pressure or your hydrostatic blood pressure, your PC, that sounds like computer, is 37. So it's a lot, right? From 120 that we start out in the aorta, the tissues, it goes to 37. Now, we have to add the pi, which is your colloid osmotic pressure of the interstitial fluid. Now, it should be always 0. What does that mean? The colloid osmotic pressure, or also called oncotic pressure, is given by albumin, the big plasma protein, like it says, plasma, should be in the plasma. We should not have any albumin in the tissues, right? This is bad if we start leaking that plasma protein. So that's what is called the osmotic pressure of the interstitial fluid. Should be 0. So we have 37 plus 0 minus this PI, which is your hydrostatic pressure in the interstitial fluid. Now, do we have to have a lot of fluid here in between the cells? No, it should always be minimal. So that's why it is 1, okay? Plus the 25, which is the oncotic or osmotic pressure of the blood plasma. So 25 never changes. It should always be 25 because it means that albumin is inside the blood vessels, it stayed in the plasma. And I'm going to tell you after this what is the oncotic pressure of plasma, but 25 never changes. So if you think about it, the 1 and the 25 are these 2 opposing forces that we have here, right? We really want to have net filtration, but we have to account for little pressure of the interstitial fluid, which should be minimal, 1. And of course, the oncotic or osmotic pressure of the blood, which is 25. And it should always be 25. Remember, this is given by albumin. So when we do our calculation here, 37 plus 0 minus 1 plus 25, we end up with an 11 millimeters of mercury as a net filtration. So it's a positive number, and that's why we have here this net filtration, right? So it's a big yellow thing telling you, yes, everything is going to go to our tissues. Julie, did you have a question?
No, I'm okay. Thank you.
Okay. All right. So are we clear on this? So imagine we started with 120 at the aorta. We get to 37 by the time we get to the tissue level. And at the end, we only have 11 milliliters of mercury. This is force, right? That is pushing things out of the capillary into the tissues. So start thinking about it. If you have a patient that has a very low blood pressure, let's say this patient starts with 90 at the aorta, do you think that he's going to get to this 11 magical number to have that net filtration and get everything the tissues need? No, right? Those cells are going to be lacking. They're not going to get oxygen. It's not enough force. There's not enough pressure to get that net filtration there, right? So that's the reason why if you have very low blood pressure, you get dizzy. You're not getting the oxygen to your brain, for example, because it's too low. There's never this magical little number here. It could be, I don't know, 7, 6. That's not enough, right? For everything to get to the tissues. All right, so now we have to analyze the other end, okay? So if you think about the other end, this is where we need to reabsorb all the carbon dioxide, all the wastes that are dissolved in that little tissue fluid, and any excess tissue fluid that is there, right? So from last week, we said in the venous end, we reabsorb about 85%. The other 15 is reabsorbed by the lymphatics. Now the lymphatics are very important because they're ultimately going to dump all that lymph into venous blood, right? So venous blood is all that blood that is returning to the right atrium so that we can reoxygenate in the lungs. Now, by the time we get to, again, we need to account for some opposing forces to that reabsorption. And here is the opposite direction, right? So you see 3 big orange arrows coming in, which is good. This is reabsorption. But then you have 2 little ones that are opposing that reabsorption. So we have to overcome that, right? So what are the forces that we have to account for on this end, on this venous end? So it's the same calculation. So you have here the hydrostatic blood pressure, right, PC, which look, from 37, it came down to 17 at the venous end. Because remember, as we go from the heart into the body, blood pressure decreases. By the time we get to the right atrium, is almost 0 millimeters of mercury. So I always visualize it in my head, like venous blood is like crawling to get back to the right atrium because there's almost no pressure by that time, right? So that's very drastic. So from 37 at the beginning of the capillary bed, we go to 17 here in the venous end. We still have to account for the osmotic pressure of the interstitial fluid. Remember, should be 0, no proteins in the interstitial fluid, minus the 1, which is the minimal hydrostatic pressure of the tissue fluid, plus 25, which is always the same and it never changes, which is your blood osmotic pressure. So when we do this calculation, if you notice, we end up with a negative 9 millimeters of mercury. Now, negative because the blood or the reabsorption is happening in the opposite. So it's good to be negative. It's in the opposite direction. We are reabsorbing. That negative there means we're reabsorbing things back into blood. So those are the forces that you need to account for. So here we have to have a good force to push things out. On the other end, we have to have a good force to reabsorb everything in. So the negative is not, you have to take it not as a mathematical negative number. The negative shows that you're taking things back in, all right? So is this clear? So the other example could be what happens when we have a patient that is hypertensive, that it has chronic hypertension, right? If he starts with 160 in the aorta, by the time we get here, this number 37 is not going to be 37. It's going to be probably 50, something like that. So having a lot of net filtration can cause things to go into the tissues that shouldn't be there. So this tremendous force of the high blood pressure is going to push things out into the tissues that should not be there. So that's the other example. With the low blood pressure, tissues don't get what they need because there's not enough force to push things out. With high blood pressure, we're pushing too many things out. And maybe protein may leak and a lot of fluid may leak. And that's the reason that hypertensive patients, most of them will have edema because this is what's happening at the tissue level. They're pushing a lot of fluid out and it's not, at the other end, we're not able to reabsorb it all because it's too much. It's over that tiny little number, which it should be 1, right? So 1, we can reabsorb, but a hypertensive patient may be pushing a lot and it's very difficult. Not even the lymphatics can take care of that. So that tissue fluid starts to accumulate and it causes then the edema. So are we good with the explanation? I know this is a difficult little slide, but as I mentioned last week, if you only know this from the whole chapter, I'll be happy because we continue talking about this when we do respiratory and more so when we start relating urinary as well, okay? So I just want you to make sure that you got it, that it makes sense to you, okay? And if you have any questions, please speak now or forever hold your peace. Yes? Are we good? All right. And somebody asked me about, do I have to know these numbers? Yeah, unfortunately, we do need to know the numbers because, I don't have my mouse thingy here, because that's the only way to remember what is the blood pressure at the venous end, what is the blood pressure at the arterial end, and that's, unfortunately, you have to know this, the numbers. I don't care if you don't know like the exact abbreviation for blood pressure hydrostatic, pi I or pi P, but that you have the concept and what the numbers mean, right? It's not just numbers that you memorize. They mean something. They mean that we are pushing things to the tissues and then we have, means that we are reabsorbing a lot, right? At the other end. So I want to go over what osmotic or oncotic pressure is. And you have this in your chapter, but maybe a little explanation on how easy you can understand this. So we have to go back to hypertonic, hypotonic, and isotonic. Do you all remember that? So a hypertonic solution is one that draws water to it. Do you agree with that? If you have a lot of solutes, you're going to be drawing water, right? Remember, water always tries to move to dissolve. So when we say that we have a hypertonic solution, it means that that solution is going to draw water. Okay, so we agree with that. So in blood vessels, and again, that line, so this is a blood vessel, we have albumin. So albumin is a very large plasma protein that should always stay inside the blood vessel. And the reason for that is that it makes the blood vessel a little more hypertonic. Now, why do I want to have hypertonic inside my blood vessel? Because I want to keep the water where it should be. You don't want to keep the water in the tissues, right? And so by having this albumin there in large quantities, is our highest concentrated protein, plasma protein, that allows the blood vessel to sustain or to keep all the water in so that we don't have leakage outside, right? Because that causes, again, edema, right? So albumin makes the inside of our blood vessels a little bit more hypertonic so that we can always draw water in. And albumin doesn't move. Albumin needs to stay in the blood vessels. So that's what makes our osmotic blood pressure. Another name is oncotic blood pressure. It's the same thing, okay? Osmotic and oncotic, same thing. So the plasma protein albumin in our blood vessels makes for that 25 millimeters of mercury. Oncotic blood pressure determines that every single little water droplet stays within the blood vessel and that we are not leaking anything out. If, and you had a discussion board on this a few weeks ago, maybe 2 weeks ago, if a patient has proteinuria, which means he's showing protein in urine, then we're affecting that oncotic or osmotic blood pressure because it's that albumin that is leaking through the kidneys and we're losing it in the urine. Well, that changes the whole picture. Now, is this patient able to get all this water inside the blood vessels? No, because there's no albumin drawing the water in. There's not oncotic or osmotic blood pressure drawing that water in. Guess what happens to a patient that it has proteinuria or losing proteins in the kidney? It has what?
Dehydration.
And what else?
Edema.
Yes. Yeah. So know how related the cardiovascular with the urinary system are, right? We have to look at the reasons why is this patient losing protein, which is mainly albumin, is a kidney thing. There's something in the kidney that is allowing this big molecule to be leaked into the urine. But at the same time, when that's happening, it's affecting cardiovascular because this patient now is not drawing enough water inside the blood vessels. The blood plasma decreases. Where does the excess fluid goes? Into the tissues, and the patient is suffering from edema. Okay, so is that clear? Because now I'm going to tell you exactly how we measure oncotic or osmotic pressure. Okay, so so here, when you think about a solution, right? And this we go back to, I don't know, chapter 6. I was just thinking that we only have 5 more weeks after this one semester to be over. So that's so much to go, right? So anyway, when we have things in solution, right? We have a solute and we have a solvent, and that is a solution. Now, most of our things in our solutes are in solution in the human body, right? Either we have them in solution inside the cells, we have it in solution in the plasma, blood plasma, we have it in solution in the tissue fluid when we're transferring things back and forth. So when you look at for example, solution of salt, the solution will be like this, right? So this is a solution of salt. Sodium chloride plus water, right? So when we think about the chemistry of a solution of salt, which is very simple, we say that I have 1 mole of sodium chloride, right? This is, we learned this in chapter 6, how do we measure concentration of a solution. It will be 1 mole of sodium chloride, 1 mole dissolved in 1,000 milliliters of water, or 1 liter of water, right? That is what we consider a 1 molar solution. But really, sodium chloride doesn't stay this way the minute that you put it in water. Sodium chloride, and you all know this because you cook, right? You put salt in water and do you see the crystals anymore? No, right? Because the minute we put water, we end up with that, right? Now we don't have a mole of sodium chloride now. We have 1 plus 1 osmotically active, particles. Sorry, particles. Okay, so that makes a difference, right? Because 2 little ions can draw a lot more water than just 1. So that's what we call osmotically active particles. And that's what gives you that osmotic pressure or oncotic pressure. So having more particles dissolved in the plasma accounts for that oncotic pressure of the blood or osmotic pressure of the blood. The more things we have dissolved in that plasma, the more that plasma is going to be willing to draw water, which is what we want. We don't want water outside the blood vessels. We need our heart to keep getting enough blood volume to pump, right? So that's what the albumin does. Not only we have sodium and chloride in the blood, we also have this albumin that, because it's huge, it cannot go through the simple squamous epithelium in the capillary. When everything is normal, the albumin, being big, stays in and maintains that osmotic pressure that is going to make sure that we draw all the water in the blood vessel. So the minute there's something wrong, either high blood pressure that will push albumin out, or kidney issues that is going to lose albumin out of the filtering blood in the kidney. The minute we do that, we're changing that oncotic pressure and we're not drawing enough fluids inside the blood vessels. And that's what causes edema, right? Because we're not taking things in the blood vessel, we're actually losing a lot because there's nothing inside the blood vessel to keep the blood plasma in. So is that clear? That's why we have to account for all these little pressures, right? And if you notice on both ends, we have that 25 and 25, right? We have oncotic blood pressure in arterial blood should be the same as the oncotic blood pressure in venous blood when everything is normal, because that albumin is what gives that little number of 25. Albumin is the one that needs to stay in there and we can keep that oncotic or osmotic blood pressure. All right. So any questions so far? No, we're good? Okay, and let me see. I think that this should be cut out. So you've all seen this picture where lymphatic vessels are blocked by a parasite. We don't see these types of conditions in our country. This is mostly in underdeveloped countries where there's still a lot of parasitic infections. People walking barefoot, and there's a lot of parasites that can pierce the skin. And this particular one likes to burrow itself in the lymphatic systems. And lymphatic systems are the ones that are reabsorbing that extra 15% or that rest 15% out of the 85. And then obviously there's a very severe edema here because all that fluid is now going back into venous circulation, right? And if you remember what I mentioned about Starling law of the heart, more in, more out, but the opposite is the same thing. If you have this severe edema and you're not reabsorbing, then your venous return is less. And by the time it goes into the heart, then less blood in, less blood out, right? And this is a cycle. So the body means the body is not getting what it needs because you have to get a good venous return in order to have a good stroke volume, which at the end is going to give you a good cardiac output, which is perfusion of the tissues, right? And then the other two things that are very important here, one is the RAS system. So RAS for R-A-A-S, RAS system, right? So this happens when, and your endocrine system will come haunt you again, because this happens when you have severe dehydration or you have severe hemorrhage, something has happened, or also when people get severe burns. There's a lot of tissue fluid that is lost when you burn the skin. Imagine taking the first layer, you get to the dermis, and that's where you have all the blood vessels. So severe burns, second, third degree burns, causes the same thing. You lose a bunch of fluid. So that causes that causes low blood pressure for sure, which in turn the kidneys are going to detect because all our blood is filtered in our kidneys. So that's why the stimulus up there, it says blood pressure is low. The arrows are not that great, but low blood pressure. And also it says low blood flow to the kidneys. So we haven't gotten to the kidneys yet, but the kidneys, we've already learned that the kidneys can detect when we have low red blood cell concentration, right, or numbers. They have these cells that can detect that. Well, they can also detect low blood volume or low blood pressure because we are filtering all our blood about 45 times a day. So the total amount of our blood, which is 5 liters, it goes through the kidneys about 45 times a day. So that's impressive. So the kidneys have this ability of detecting if there's not enough blood flow to the kidneys. The kidneys are not only detecting that, but obviously they're getting rid of the soluble waste. If the kidneys are not getting enough blood flow, they're not going to be cleaning the blood as well. And you're going to be building up toxic waste in the blood, right? So that's why kidneys are so related to cardiovascular, right? So when that happens, when the kidney detects this low blood pressure and low blood flow, these little cells that are in the, it's called the juxtaglomerular apparatus, and we're going to learn that that is a group of cells that are a little different, and they're very close to where we do filtration of the urine and where we do secretion. So for now, just stay with that. But when these little cells detect that, they're going to secrete a hormone that is called renin. So here is your little renin, right? And renin is kind of like an enzyme. It's really a hormone, but it's an enzyme to catalyze the conversion between angiotensinogen and angiotensin 1. Now, if you, let's see if I can do this. Nope. Here. So just want to do this and I can't do it. Okay. So angiotensinogen is in the liver. We have it in the liver. Angiotensin 1 is in the lungs. Now, don't ask me why. That's how we are wired. We have angiotensinogen in the liver and we have angiotensin 1 in the lungs. So renin goes to the liver converts angiotensinogen into angiotensin 1. Angiotensin 1 then goes to the lungs, and with this little enzyme that you see there as an ACE, ACE, it becomes angiotensin 2. So what is ACE or ACE? Is angiotensin converting enzyme. That's the name. For short, we call it ACE. Now, why this conversion of angiotensin 1 into 2 is in the lungs? Because that's the only place that we have ACE, that enzyme. That weird? Yes, very weird. So angiotensin 2 goes to your adrenal cortex, and it's going to make your adrenal cortex secrete aldosterone. Aldosterone, major action, was to increase sodium reabsorption. So with sodium, remember we have salt is sodium chloride. Anytime that you hear salt, you have to imagine that water goes with it. So because we're reabsorbing sodium chloride because of aldosterone, water is going to be reabsorbed as well, and that's going to cause the blood volume to increase and increasing the blood pressure as well. In the meantime, too, when we have angiotensin 2, it causes vasoconstriction of the arterioles. So vasoconstriction means reducing the diameter of the arterioles, which overall also increases blood pressure. Okay? So this is what happens when you have severe dehydration, severe bleeding, major burns, where we affect the blood volume, the blood pressure, so the RAS is put into place. So is this clear? So remember, aldosterone was a mineralocorticoid, right? From the zona glomerulosa of the adrenal cortex. And the major function was to reabsorb salt in the kidneys. Therefore, anytime you see salt, water goes with it. Same thing if we say salt is being excreted in the kidneys. If you hear that, if you're getting rid of salt through the kidneys, water goes with it as well. Okay, so always have those two together. Just think, if you eat salty foods, you're thirsty. So there's always a relationship between salt and water. They go together. Either way, reabsorbing or getting rid of, right? So kind of like water in this case is a secondary action of aldosterone. Mostly it's the reabsorption of sodium, but water goes with it. And that helps to correct the initial stimulus, which was the low blood pressure, low blood volume. So when we look at the opposite effect, right? When you have increased blood volume or increased blood pressure, right? So when you have increased blood volume, that will give you an increased venous return. More in, more out, right? Less in, less out. So having high blood pressure, high blood volume, for sure you're going to have an increased venous return. So when you have a lot of blood returning to the atria, there are a little group of cells that are going to detect that because the atria are going to stretch. And that stretching causes the secretion of this little hormone, which is called atrial natriuretic peptide, or for short is ANP. So this ANP, what causes is increased salt excretion. And remember, sodium chloride goes with water, so we're going to get rid of salt and water through the kidneys, So we're going to have a higher urine volume, which is going to make us reduce the blood volume. So this is the exact opposite case of the RAS, right? And other things that happen is that the stretch of the atrium causes the vagus nerve to send information to the pituitary to do not secrete antidiuretic. I know it doesn't make sense, but I don't like the arrow saying decretion of antidiuretic. It's better just to say no secretion of antidiuretic. Because remember, antidiuretic is only secreted if we have low blood volume. That causes us not to make a lot of urine, right? But in this case, you already have a high blood volume, so you don't want any ADH to be secreted, so that we can get rid of salt, get rid of water, and that controls or restores our normal blood volume. So you have these two cases of hormones that are involved in helping us with maintaining blood volume. In any case, any spike or any drop in our blood volume and blood pressure, we can rely on those hormonal groups to do this. One thing that I forgot to mention on the RAS system and that little ACE, because we're going to see it again in respiratory. There's a lot of cardiac patients that take, we mentioned beta blockers, but the other treatment is ACE blockers. And they're blocking this little enzyme. And why would they do this? Cardiac patients, they already have high blood pressure, right? So you don't want to increase it even more with aldosterone. So some treatment will be just to block that ACE enzyme so that they don't do this, so they don't increase blood volume and blood pressure. What are the things? And I know that Chris is gonna ask, so if you're skipping slides, we don't have to know it. Yeah, you have to know. I'm just skipping because we are in class and I cannot possibly go with through everything. So this is, obviously you all know, and it's nice to know what happens when we are exercising, right? And if you think about athletes and things like that, they are very efficient in the way that they pump blood and oxygen to their lungs, to their skeletal muscles, but they're also very efficient when they do that exchange in the lungs. And also that's why the heart in athletes is a little more enlarged, right? So all those things, why I cannot see the chat? Come on. Oh my God, okay. Yeah. Yes, Celita, you can also get edema with RAS. Yes, I couldn't see the chat without going away from the PowerPoint, sorry. All right, so what is vascular resistance, right? This is at the beginning of the chapter, I think, when we talk about arteries and veins. And let me do it in my whiteboard. Oops, stop. So when, okay. So when you have a blood vessel and the blood flow comes, oh, sorry. And the blood flow comes here, blood flow, right? Blood pressure is the interaction between this blood and then the opposing forces of the walls of these arteries, right? So the blood pressure needs to overcome these opposing forces from the wall. So the opposing forces from the wall is what is called resistance. So I have to have a certain blood pressure so my blood flow can go through that opposing resistance. Yeah, so that's blood vessel resistance. The wall creating these opposing forces to the blood flow. So the blood flow has to have a specific blood pressure so that we can oppose the resistance that the wall is giving the blood flow. Is that clear? So when you have a very large aorta, this should be continuous, right? This is the diameter of the aorta, big, right? Most of the blood flow is here. And this big diameter doesn't offer a lot of resistance, right? So in big arteries, blood flow is fast and it goes with almost no opposing forces because the circulation of the blood is kind of like concentric here in the middle. But if you think about a little capillary, right? And so a capillary this big, and you have the resistance in this capillary, imagine that that's going to be a lot of resistance to that blood flow, right? So big arteries, low resistance, is inversely proportional. Small little capillaries, high resistance, yeah? So when we think about total peripheral resistance, what does that mean? It means that we have to add up all the resistance of all the little capillaries in our body. That's huge. That's huge because your tissues depend on this total peripheral resistance, right? Your tissues are waiting for blood flow to get to them. And if you have a very high total peripheral resistance, that makes it hard for the tissues to get everything. So that's why we have to account, right? The other thing with blood flow is the thickness of the blood. Some people have polycythemia, it's called, right? You have oops, it is poly like that. This is increased red blood cells. So that makes your blood very thick. So again, if you have a very thick blood and you're trying to circulate through these tiny, tiny little capillaries, imagine the resistance there, right? It's not going to be enough to push through that resistance if your blood is very thick. So the way to remember this is drawing honey through a straw or a big one that is this wide in diameter, right? But be a lot faster to draw honey up in the big one than in a tiny straw. So that's what happens in our capillaries. So they're good because they have one cell thick and we can send everything to our tissues. But again, they depend on your blood pressure at the tissue fluid because they have a lot of resistance, right? So we have to account for that as well. So all this just so that we can go back to this. So arterioles have high vascular resistance when they are at rest. And that is because they have a lot of muscle, right? Different than your venous circulation. Veins don't have a lot of muscle. And if you notice, the veins don't have a heart at the bottom of the feet, right? We don't have another unit at the bottom of our body that is pushing venous return back. So the only way that we can keep venous return going is because we have some valves in our veins that are kind of like pushing. The valves are little flaps of tissue on the inside of the endothelium that they go up, and those are the ones that are helping to send the blood back up to the superior and inferior vena cava. And that is also aided by skeletal muscle. So skeletal muscle in our legs helps with venous return. So if you notice people that are on their feet a lot, or even I do it when I teach face-to-face, I walk a lot because teachers, we are on our feet a lot of hours, right? So blood tends to pool in your legs. But if you move, right, your skeletal muscles help to return that venous blood back up, right? Because we don't have another pump. So when you are exercising at rest, we receive about 20 to 25% of the total blood supply to our body, right? And when you contract the muscles, the blood flow decreases because you're contracting the muscle. It's almost like you're doing a vasoconstriction to your arterioles, right? So epinephrine and acetylcholine, sympathetic, will vasodilate those little arterioles that they tend to stop or to decrease the diameter when you're exercising. So when you're exercising, you have some epinephrine, right? It's the euphoria of the exercise, and that allows for a little vasodilation to the skeletal muscle arterioles so that they can have more blood flow to bring more oxygen to continue doing muscle contraction. So what happens in dynamic exercise, and that would be like running or doing strenuous exercise, you try to decrease the vascular resistance through the skeletal muscle because obviously you're increasing cardiac output, your heart rate increases, so your stroke volume increases, cardiac output increases. You have that vasodilation from the epinephrine, and also you're now sending blood to other organs in the body. So we are diverting blood from other organs so that we can fully supply our skeletal muscles, right? It's what I said. If you are exercising and you stop to eat, that's not a good thing because you're diverting from the skeletal muscles blood to that food that you just put in your stomach, because your body is going to think, okay, she's exercising, but now she's putting food in the stomach. I have to go there and try to digest it. So it's not smart to be eating at the same time that you're exercising. It's, you eat before or you eat after, but not during, okay? And I mentioned this also, only if you're running a marathon and you're just doing a little of those packets, which are pure glucose, really, to keep you going, because this is a marathon, right? But when you're normally exercising or taking a class and things like that, don't eat. You don't have to eat. Oh, look at this one came with the ducky. So when you have endurance training, and these are athletes, they have a very good oxygen delivery. They have a lower resting cardiac rate, right? They are able to train so that they slow down the spontaneous depolarizations that the SA know. They have a smaller, they increase the stroke volume because they have an increase in the blood volume. But again, more blood in, more blood out. The heart needs to contract harder. The heart needs to contract stronger. So that's what makes athletes have enlarged hearts at the end, right? So they have a wonderful endurance. They have a wonderful oxygen supply. But at the end of their careers, most of them will end up with enlarged hearts, right? And that's not that great. It's a muscle. So if you work a heart, it's going to increase like any other muscle, right? Even skeletal muscles. And so this is how the body redistributes the amount of blood. Remember, we don't have a lot of blood to go around. It's only 5 liters. And you don't have to memorize these numbers. You don't have to know all this. But what the body wants to do when you're exercising, and just look at these little circles, is that most of the blood is going to go to the skeletal muscle, and we're going to decrease the supply to the other organs, right? Because we're not needing those organs. You're not going to be studying while you're having heavy exercise and all that. But if you look at comparison at rest and look at these little circles, right? They're pretty even how we distribute the blood, right? But the minute we start exercising, we have to divert most of the blood to the area that is busy at that time. See, 5 liters of blood total doesn't allow for us to give every single organ the right or the same amount of blood. It has to be taken away from some organs and given to the organs that are active. So that's what happens in this is just the outline of everything that we've seen. With exercise, athletes will have increased cardiac output, and that comes from increased cardiac rate, increased stroke volume, right? And that allows for increased blood flow to skeletal muscles, which in turn, remember that skeletal muscle activity causes vasodilation in those little arterioles and sympathetic contraction of the other organs in the body. Because you have more blood flow, you increase your venous return and you continue the cycle until you stop exercising, right? In the cool down, all that is reverted, right? We slow our heart rate, we slow or decrease our stroke volume, we decrease our cardiac output. There's not a lot of blood going to the skeletal muscles. Oh, this is important. The baroreceptors. So we learned baroreceptors, I think it was chapter 10. So baro means pressure. So we have these little baroreceptors in the carotid artery, the common carotid, which is the major one that sends blood to the brain. And we have it in the aortic arch. So these groups of little cells, they're called sinuses. So there's kind of like a little depression in the common carotid, and in the aortic arch. And that's what these baroreceptors are. These are specialized sensory neurons, right? So these little cells are detecting blood pressure. So obviously, why we have it in the carotid and why do we have it in the aortic arch? What do you think? Why they're not anywhere else? Blood is coming out of the left ventricle through the aorta, right? And what is the systolic pressure there?
120.
Is the highest, right? So of course we want to have those sensors right there, those baroreceptors, because the blood going through the carotids, if it's too high, they might burst cerebral little capillaries, right? And that will cause a cerebrovascular accident. And same with the aortic arch, right? If the blood comes out with a lot of force, we may be causing issues coming down to other of our capillaries in the body. So these baroreceptors are sensing, right? So we go back to the reflex arc, right? You have a stimulus, which in this case will be increased blood pressure, right? So we have that stimulus. So the receptors are the baroreceptors. Those receptors are saying, hey, blood pressure is too high here. So they send that information to sensory neurons, and then they send it to the cardiovascular centers. That will be the interpretation center, right? That we learn in our reflex arc. So these interpretation centers, they're called the cardiovascular centers. They're in the medulla. So that's why you have this here, medulla, right? So there's two kinds of cardiovascular receptors. One is the one that detects blood pressure, and the other detects vasodilation, vasoconstriction. The dog is going nuts. All right, so right now we're talking about the baroreceptor reflex. So we sense that this blood pressure is a little high. So you see how the information is going from those receptors to a sensory neuron in the interpretation center. They're both going to the same way. And what do we have to do in response to that? If the blood pressure is high, then we're going to send the parasympathetic response to the SA node to slow down heart rate. And that's going to take care of high blood pressure. Remember, high blood pressure, high heart rate, high blood volume, right? So the sympathetic, the parasympathetic, the vagus is cranial nerve number 10, which is the only cranial nerve, remember, that comes out of the brain and goes down to the heart. And then we're going to see that it keeps going down to the internal organs of digestion. So the vagus is a very long nerve and is parasympathetic. So that one is going to put the brakes on the sinoatrial node to slow down heart rate and control that spike of blood pressure. And that happens all the time. And this, we're talking normal people. Okay, who's normal? Let's say people that have normal blood pressure, right? Because it could happen that you're exercising and your blood pressure goes up. That's normal because your heart rate is increased, right? But your baroreceptors know what to do. They're gonna sense that, and what they're gonna do is they're gonna vasodilate the blood vessels in your brain so you don't drop from a CVA during exercise, which has happened to people, right? When you get the news, for example, he was on the treadmill and he dropped dead of a heart attack. You've heard that, right? It had happened. Well, these are people that, first of all, didn't ask the doctor if they could be running like maniacs on the treadmill. They didn't know they had high blood pressure. When you have high blood pressure chronically, your baroreceptors are so used to that they don't respond anymore. And you start exercising like crazy, you increase your heart rate, you increase your blood pressure, your baroreceptors are used to high blood pressure, you increase it even more during exercise, boom, you burst the little capillaries in the brain and you can die right there on the treadmill. So how important it is to know your blood pressure, to know your genetics, to take care of yourselves. And if you're gonna start exercising when you haven't done it for a long time, it's better to be safe and check with the doctor. How fast do I have to run, or how strong do I have to start my training? Don't start like a maniac running for an hour on the treadmill when you don't know anything about your blood pressure, your heart condition, and things like that. And it could happen to young people as well, right? Because as I said, genetics have a lot to do with your blood pressure. So you have to know what was the blood pressure for your parents, your grandparents, and how you cannot change genetics, but you can change the expression of your genes according to how you treat yourself with exercise and nutrition, right? So my dad died of a massive heart attack at 67. He had chronic hypertension, and his parents both died in their 60s as well. And his brother died at 48. So imagine the genetic charge that my sister and I have, right? We know that we don't take care of ourselves. We could have that genetic. Luckily, my sister and I think, took after my mom, which used to have the lowest blood pressure in the planet. So we are pretty good. But because of my genetics, I take care of myself. So I eat very healthy. I check my blood pressure, and I do a lot of aerobic exercise, like my tennis, because I have to really check my heart and my blood pressure. So we all should do that, right? So we don't have those surprises on the treadmill. Yes, Julie.
I'm sorry, I'm trying to remember, and I should probably know this, but you said there's two cardiovascular receptors. One is for blood pressure and the other?
It's called the vasomotor center, which is for constriction and dilation of the vessels.
Got it, thank you.
They're both activated, right? The baroreceptors detect blood pressure, which are going to affect heart rate. But at the same time, if you have high blood pressure, the vasomotor center will vasodilate to even help even more with lowering the blood pressure. Oh, and it's here. I was talking about that, and it's here. So safety happens when you go from laying to standing. This is how the body can compensate. And a lot of times you're going to hear in cardiovascular, what does the body do to compensate for this, for a specific stiffness? So this example is when we're laying down and you get up very fast, right? Sometimes you get dizzy. And so immediately the heart is going to compensate so that we don't get dizzy or we don't pass out, right? So when you go from laying to standing, because of gravity, you have a decreased venous return. So that's the whole reason, right? Remember, venous return is all the blood that is coming from the bottom of your feet and the top of your head back to the heart, right? But when you're laying down, blood pressure is the same at all parts of your body, right? Because we don't have to fight gravity. The blood flows and comes back. We're the same. The minute you get up, all that blood pulls down, and that causes a reduced venous return, right? And again, if you reduce venous return, so you have less blood coming in, you're gonna have less blood coming out, decreased stroke volume. And that causes decreased cardiac output. Stroke volume and heart rate, cardiac output decreases, you have a low blood pressure. So that's what causes the baroreceptors, and all that happens so fast. The minute you're laying down and you get up, all this is happening. We don't realize that, right? So when that low pressure hits the baroreceptors go, uh-oh, this person is gonna pass out. I better do something. So the baroreceptors detect a very low blood pressure. It sends the information to the cardiac center to increase cardiac rate, and at the same time to the vasomotor center to vasoconstrict the arterioles, and those two are going to increase the blood pressure. So if you don't notice anything when you're laying down and you get up, it's because everything is working really good in this example. Your baroreceptor reflex is perfect. Now, if you notice that you get dizzy, your blood pressure may be really, really low, and it takes a little bit for all this to happen, all these reflex to happen. But if you don't notice when you're laying down and then you get up and you're fine, all this has been going on, right? Because the baroreceptors and the vasomotor center, they are detecting everything that is going on. But again, normal blood pressure individuals, right? If you have constant low blood pressure or constant high blood pressure, the reflex doesn't work that well. Yes, Julie.
So when you're actually experiencing dizziness and say like you even get like stars or your vision kind of gets blurry, that's a lack of blood supply to your brain that
yes,
quickly.
That is one of the reasons. The other reason could be low blood sugar, right? Hypoglycemia. So if you haven't eaten in a while, that could also cause the dizziness.
From standing. So if you have
yeah.
low blood sugar and then you stand, both of those are not a good combo. Okay.
They're not a good combo. I used to have a colleague, she retired. She was teaching anatomy and physiology, and she retired already, but she always will say, I'm hypoglycemic, I'm hypoglycemic. And I kept saying to her, well, eat something. Because she kept saying that, but she every day. I mean, if you know that you are a little bit hypoglycemic, you have to always have something with you, right? Because she was teaching all day and all night. She was a maniac. And, but she always would say, oh, I'm low blood sugar. That's why I'm passing out. But anyway, she's happy now, retired, I guess. So here's the blood pressure, which we already did with our labs last time. And what else is here? Let me see. Oh, this is good. No, it's not good, but yeah. So we have what is called shock. So different types of shocks, but I'm sure that you've heard in medical shows or things like that, the patient is going into shock, right? Shock means you're not giving enough blood flow to the tissues, and that means no oxygen to the tissues. And of course, the body, the minute that happens, the heart will start to compensate, always compensate. So when there's not enough blood flow to tissues, the heart will try to compensate, but sometimes the heart will overdo it and that can lead to death anyway. And by overdoing it, I mean, if there's a loss of blood going to the tissues, the heart is going to start beating really fast. So it's going to be an increase of heart rate, and that itself can cause a death, right? So the compensatory mechanism for this is to increase the heart rate, but at the same time, if it's really bad and the heart rate is really high, that leads to death anyway. So what are the signs of shock? Obviously, low blood pressure, right? There's no pulse pressure. You have a decreased volume of urine. That tells you blood is not going through the kidneys because you're not making any urine, right? So that's a sign. Your blood pH increases, so it goes very alkaline. You're going to see this in respiratory. We're going to learn that. If you hyperventilate, you go into alkalosis. Hyperventilation means that you're getting rid of a lot of carbon dioxide fast. And that happens at the beginning, right? If a patient has very low blood pressure, patient starts to breathe very hard, and that's what hyperventilation does. And then obviously you start to have dry skin, or they say restlessness. You start moving because you're not having enough blood flow to the tissues. So you don't have increased perfusion, the word that I love, right? Now, at the end of the shock, and this is very serious, you start having no urine volume, decreased systolic pressure, so you can't take a pulse. Then you go into acidosis, which is the opposite of alkalosis. Your respiratory rate is very shallow and very low, like slow, and your body starts to get clammy, cold, and obviously it says cloudy senses because the person doesn't make sense. So this is at the end when somebody's ready to die. So what does the body do to compensate for a circulatory shock? It could be because of hemorrhage or something like that. Your heart starts to increase, your heart rate starts to increase tremendously, okay? And also it will increase the contractility, the strength, okay? Then at the same time, we're gonna reduce blood flow to the rest of the organs. So you see digestive tract and skin, you have decreased blood flow to all of them. And you're gonna have a decrease in urine production or urine output. The posterior pituitary is gonna secrete ADH so that we can conserve as much blood as possible, because circulatory shock means you lost a lot of blood volume. So the hypovolemic shock is very similar, because this is where you have lost blood because of injury, right? Dehydration, or like I mentioned before, burns. Remember, first or second or third degree burns means that they taken off the first layer of the skin, and that's where all your, in the dermis is where all your little blood vessels are. So you lose a lot of fluids that way. So when you don't have enough blood, decrease cardiac output, decrease the blood pressure, and the compensatory mechanism is to increase heart rate. So remember, increasing heart rate, the compensatory mechanism is good, but it could go all the way, and it could increase heart rate so much that the person just dies of a heart attack. Then you have septic shock, and this happens when people have massive infection, but this is where the infection has reached the blood. So that's called sepsis, right? And people can die very, very fast. That's why it says mortality rate is very high in septic shock, because all these toxins of the bacterial infection starts to go everywhere, and it causes what is called massive vasodilation or systemic vasodilation. So we like to have control vasodilation and vasoconstriction, This is how we divert blood flow to different parts of the body. In some areas we vasodilate, in some areas we vasoconstrict. But when it happens systemically, and think about every single little artery is vasodilated, the blood pressure drops tremendously, and that's what causes death. We also have anaphylactic shock. This is a serious one, and this is where histamine, again, is released systemically. So when histamine is released locally, like we're gonna see in immune system, it can cause vasodilation to a little area that is damaged. And with the vasodilation caused by histamine, you bring more blood flow to the area to heal. But when it happens in anaphylactic shock, it means that histamine is secreted systemically. And again, it causes that massive vasodilation, and it can cause death as well. So somebody that has severe allergies, right? That you need to have your EpiPen or something, because anaphylactic shock can be very dangerous. I mean, it kills people. And then you have congestive heart failure, and this is a chronic condition for some of the cardiac patients. It happens because of a myocardial infarction, which is heart attack. The myocardial infarction causes areas of the heart tissue to die, to be necrotic, and they cannot contract the same way. So if the heart has several areas that are damaged, they're not going to be able to contract and send blood to the rest of the body. And it can also be caused by hypertension, and this is chronic hypertension, again, because hypertension causes the body to handle high blood pressure chronically, and that causes the heart to work harder to try to push all that blood out. Remember, more blood in, more blood out. In hypertensive patients, the heart, as it enlarges, instead of being stronger, it starts to affect the contractility of the heart. So instead of, because it's larger, contract stronger, it's larger but starts to contract weakly. So instead of trying to push all that blood that is coming in out, because it's contracting with less force, some of the blood starts to back up, and that's what happens with congestive. The word congestive means backing up, and in the place that backs up most of the times is in the lungs. So that's why cardiac patients that have chronic hypertension, they also suffer from congestive heart failure. It's a condition that can be treated, but it cannot be cured. And most of the time, patients with congestive heart failure, unluckily, but they find death for sure. So you can have congestive heart failure from the left side, they call it. This is the one that causes the pulmonary edema, which is the congestive lungs. Or you can have right side failure, and that causes systemic edema. So backing up in the lungs is the left, backing up in the tissues is the right side. Neither is good. They're both congestive heart failure. And this is very important. Heart failure is that at the end, these patients die because the heart just fails to keep contracting. It's very weak in their contraction.
For the left side, are you basically drowning?
Yes, yes. That's what people say. Oh, he drowned in his own blood. Yes. When you have that congestion in the lungs are not really functioning like they're supposed to, right? Again, and the oxygen and carbon dioxide is not being exchanged the same way. So yes, that's what people say. All right, I've been talking for an hour and a half. So let's break for about 10 minutes so that we can go over our labs. And I don't know if I'll have time to go over chapter 15, but since you had the exam on 12, 13, and 14, I thought that it was important for you guys to kind of like reinforce some of the concepts which are very important. All right, so we'll stop for about 10 minutes, okay? So the first lab that we are going to see is ELISA, and that means enzyme-linked immunoassay. It is the typical test where we can find out if a patient has been exposed to a specific antigen. Antigens are anything that is considered non-self, right? Because you remember when we did blood typing, those little flags or antigens on the cell surface, those are considered self antigens, meaning that we're born with those and your immune system recognizes them as self from the body. But anything that is non-self, bacteria, virus, parasites, or cells from another individual, those are considered non-self, and your immune system is going to mount a response trying to destroy those. So that's why when we have organ transplant, you need to match the patient, the recipient with the donor as close as possible so that we don't have these immunoresponses. Because if the organ is not matched the perfect way, then your immune system is going to reject that organ, it's going to attack it. And that has happened where a kidney has been attacked by the immune system and needs to be taken out of the body and replaced by another organ. So luckily, when we have those type of situations of organ transplant these days, they can match up to 75% of those self antigens that we're born with. Obviously, if you have an identical twin, that would be the best donor for an organ. But because not all of us do, then they can match the little surface antigens, the ones that are self that we're born with, between the donor and the recipient. And if 75% of those match, then they go ahead with the transplant. The other 25% is taken care with immunosuppressant drugs that transplantees need to be on for many, many years, and sometimes forever after they have an organ. So ELISA is very common in many labs. Some labs, that's all they do is just do ELISA. You just need little blood plasma, and all you can tell is that the patient has been exposed. You cannot tell if the patient is sick, because you can have a lot of asymptomatic patients. And we've seen this with COVID. A lot of asymptomatic patients can transmit the disease, and even though they don't have any symptoms. And in other times, ELISA can give false negatives. So every time that you do it, you have to do it multiple times. So you do it with dilutions and several times for the same patient, because it can give false negatives. And also some individuals, they're not very good antibody producers. I don't know if you know, but the COVID vaccine, for example, protects you about 80% after the first shot, and then about 95% after the second shot, but only after 15 days after your second shot is that you're considered immunized. And again, because it's 95%, that means that 95% of the time, if you get it, you're not going to die. It's going to be mild. But there is that 5% that we're not covered. But of course, it's a lot better than not having it, right? But there's a lot of asymptomatic people that can be carriers of a disorder or disease, and they can transmit it, but they don't have any symptoms. So our next It's a little video on the two kinds of enzyme linked immunoassay or ELISA that it can be done. They're called direct and indirect. So the direct is the one that checks for the antigen. So they're specifically looking for a specific antigen, let's say COVID-19 virus. Or the indirect is the one that looks for the antibodies to that specific antigen. So let's see if my dog stops barking. Oh, come on. Why I can't do it? Huh. That little video is cute.
Okay, you might have to get into the presentation mode without notes on the side, and then I think it'll play.
Yeah, exactly. That's what I'm trying to do. All right. There we go. So what you see, most of the labs, they will do the indirect type of ELISA, where you are looking for a specific antibody. So you know the antigen that you have to lay or cover the wells with. So the first step is to add that antigen, the known antigen, to the well. Then you add the patient's serum, which has the maybe antibodies to that antigen. And then you have to add a secondary enzyme-linked antibody that is going to recognize antigen-antibody complex. So this secondary enzyme-linked antibody is so that we can visualize the sample, because we have the antigen that we put first, the plasma with the possible antibodies of the patient. But if they match, if the patient has the specific antibody, we can't see it because it's colorless. So this secondary enzyme linked is helping us to visualize it, because when we add the substrate to that enzyme, then now we know if it changes color, is that we have the original antigen antibody attached. If we don't have any color, it means the patient didn't have any antibodies, and every single of the steps did not attach to each other and the color didn't change. So a change of color means the patient has the antibodies, and if we don't see a change of color, the patient doesn't have the antibody. Like I said, having a negative ELISA is not 100% foolproof. It could be that the patient has very, very small amounts of antibodies and they're not really enough to be detected. Or it could be a error in making the dilution of the sample. That happens as well. So that's why it has to be repeated within the same plate, which is called the microtiter plate. You have to repeat it several times for each patient so that you make sure that you're doing it right, or that that you have a good repetition, so you can be sure. So any questions with ELISA? We used to do it one by one in our labs, but that's the simulation is all I could find. Yes, Julie.
Would, I feel like it's a trick question. Maybe I'm overthinking it. Would HIV be different or the same?
No,
Okay.
same. We do it for HIV, we do it for rheumatoid arthritis, we do it with Lyme's disease, you do it with hepatitis A, B, C. Yes, it's for everybody's the same. So again, antibody titer means concentration of antibodies. So when you do the ELISA, you will have a dilution for each patient. So the darker the color means the more antibodies you have. So if you see it in all the dilutions, it means this patient has a lot of antibodies. The amount of antibodies or the titer has to do with the time of infection. If you have a lot of antibodies, it means that the infection has been more recent. If the antibodies are not, or the titer, like it's properly called, if the antibody titer is not that high, it means that the patient maybe has been exposed a long time ago. So in our body, the antibodies, they can last, depending on the antigen, but they can last about 20 years. That's what they don't know about COVID. They don't know that we might be needing like a booster every year, like we do with the flu, to make sure that we keep having that constant titer in our blood of the antibodies. And the main function of antibodies, I know that we're not really covering chapter 15 just yet, but the main function of antibodies is what is called to neutralize the antigen. So if the antigen is floating anywhere in your body, having the antibodies is going to be recognized immediately, and the antibodies are going to surround the antigen or the infected cell, which is how the viruses infect, they get into a cell. So the antibodies are going to surround the cell that is infected because they're going to recognize that something is not right. And by doing so, they're going to make it more visible to the T cells, and the T cells then can come and attack by direct contact. So T cells are going to then attach to this infected cell that has been kind of like tagged by the antibodies, and they're going to poke holes on the cell, and the cell is going to be destroyed or lyse. So that's basically how antibodies work. So B cells produce antibodies, and then they're going to be circulating in our plasma, kind of like policing. And then the T cells are the ones that kill by contact. So we need both. We need both type of immunity in order to mount a good immune response, right? It's first the entrance of the pathogen, the developing of the antibodies, and then the attack by the T cells. So we need both. We can't just have one, right? But in the case of HIV, T cells are the ones that are attacked by the virus. So that's why it's so dangerous, because the very same cells that are supposed to be killing by contact, those are the cells that are being invaded by HIV. So that's what is a little tricky with HIV. So what they've done with the new medication cocktails, people now can live long lives, almost normal lives, because the cocktail of medication has reduced the viral count in these patients to so low that they have very few T cells infected. So that's how it's been great. They couldn't really cure it, but having such a low count of virus in their body makes it that their immune system is still going and is still strong and being able to fight other infections. At the beginning of HIV, the patients will be dying from secondary infections, like you've heard that they will develop carpoxysarcoma, which is a skin cancer. They will develop fungal infections that were very serious, and people would die of those because the immune system was so depleted by the virus. But these days, they figure by keeping the viral count really low, the immune system still is strong enough, and these people can live almost normal life. Yes, you had a question, Julie?
I was just going to ask about the other process that you mentioned, unless we're about to go over it. Maybe I jumped the gun.
No, the direct ELISA?
The one where it's actually searching for antigens instead of antibodies.
Yeah, we don't do that one that much because you have to, well, they're doing it now with COVID because they know what they're looking for. They're looking for COVID. So they do that antigen test, which is a lot shorter in time than the antibody one. And it's a lot shorter than the PCR one, right? The COVID testing. So they started doing the rapid test, they call it, and it's looking for the antigen. So that would be cases where you know exactly what you're looking for. So you look for the antigen, and that's where the what we add is the antibody against the antigen.
Okay, so in that case, then you know that the person, if they are positive, they actually have it. It's not maybe a past exposure.
Right.
They are currently exposed.
Right, they have it, yes. They have it in their body. So it could be without symptoms, but they have it. So if they have it, they can pass it on, right? They can transmit it to somebody else. So that's why with the vaccine now, and I don't know how you all feel about the vaccine. I know that some of you have gotten it. But my sister, for example, she doesn't want the vaccine because she thinks that they're going to get into her DNA and all that stupid thing. And my sister is a dentist. So I had a fight with her because I said, you're a scientist as well. You know what I'm talking about. And she says she doesn't want to have a vaccine. But anyway, we have those people in this world. And I even have it in my own family. So in order not to fight with my sister, I just say, I respect you, but I don't believe what you're saying, right? But that's why they say that even people that are vaccinated, they could still transmit it if they get it. You're not going to have the symptoms, but if you happen to have it, you may pass it on to somebody. So that's what my sister says. If you have the vaccine, then why have it if you're going to pass it on anyway? That's her reasoning. But I say, yeah, but I know I'm not going to die. Right? That's what I'm doing it. I'm getting the vaccine. So if I get it, it's going to be mild. It's just like the flu shot. I get it every single year because I'm in contact with all of you students when we're face to face. And I get it because, yeah, sometimes I'm sick, even with the flu shot, but I'm not in bed for 2 weeks or high fever or even worse, right? When you get a certain age, flu can kill you too. So, I think I take COVID the same way. It's just so that if you get it, it's a mild case and you can survive it. That's it. But okay, my sister doesn't see it that way, and she says, "You do it, and then I'll see if you have any negative side effects, and then I'll have my vaccine." And I say, "Well, the side effects may not appear like for 20 years, so I don't know what to tell you, right?" Okay. So, any questions on ELISA? Very appropriate for these days. By the way, my sister retired, so she thinks she doesn't have to see patients or anything, so she doesn't care. All right, so we have two little virtual labs on respiratory system. And I don't want to get too involved in respiratory because I want to flood your mind with new information. And I know that you have to study those other 3 chapters for the exam, but a little bit so that we understand what we're measuring here. So breathing, of course, is taking an inhalation so that we can fill our lungs with oxygen. And from what we learn in the cardiovascular, you know that in the lungs, once you inhale, that's where the venous blood is going to be coming in, and you're going to be doing that exchange. The blood is going to be charged back with oxygen, and that carbon dioxide that we brought to the lungs is going to go the opposite route, and we're going to exhale it out. So obviously the respiratory rate and the cardiac rate are totally related, because every time that we inhale, that venous blood that is charged with carbon dioxide is already in the lungs so that it can do the exchange and immediately get rid of the carbon dioxide. So taking air in and out is a very physical process. There's no magic here. It's just a change in pressure gradients. So remember that a gradient is a difference. So it's just with our respiratory muscles, which is the major one is the diaphragm, that is at the bottom of the thoracic cage, and the intercostals, external and internal, in between the ribs. Those are our major respiratory muscles. So when we inhale, we make our thoracic cage bigger. The volume gets bigger. So if you put your hands in your ribs and you inhale, you're going to see that not only your shoulders go up, but the ribs go out. So we're making our container, I call it, larger. So at that moment, when we inhale, making our container larger, the pressure inside decreases. So the pressure inside is less than the pressure outside. So when we learn about diffusion, remember that we can go from high to low with no problem, right? If that's downhill or downgrade, right? Or downstairs. So this is the same concept. The minute that we make our inhalation and we make our thoracic cage larger, the pressure inside our lungs is smaller or lower than the pressure outside. So what is the pressure outside? The pressure outside is our atmospheric pressure, right? So at sea level, it's about 760 millimeters of mercury. So when we inhale, the pressure inside our lungs is 758. So then we have high pressure on the outside, low pressure on the inside. The air just flows very easily from high outside to low inside. So that's what happens in inhalation. So it's the same concept of diffusion from high to low, right? So making the container larger decreases the inside pressure, and now we have higher pressure, atmospheric outside, lower inside, and that's how we take air in. So what I said is a very physical process. It has to do with laws of physics. Yeah, Julie, you had a question?
I did. Okay, so you said sea level outside is 760, and when we inhale, it's 758 within
Inside.
our lungs.
Yes.
What is it starting before we inhale?
Same. If you don't breathe, you're dead. So it will be zero. But
well, you said the pressure is higher before we inhale, right?
no,
inside.
It will be the same. Think about that you're at the same because you're sea level, right? But the minute you inhale, you change the size of your container. So that's when the pressure goes lower than the one that we have outside.
I see, okay.
Yeah, so you can think about it like it's the same if you're not even breathing, but the minute that you inhale, making all this bigger, it lowers the pressure and the air just flows in. So when I said it's a physical process, it has to do with physics laws, right? So I told you that I have a biochemistry undergrad major. So when I was studying my major, I said, why do I have to study physics? I don't care about physics. I only care about biology and chemistry. And my physics instructor, I remember, said, you'll see that you use it sometimes. And I'm like, never in my wildest dreams. And of course we use it a lot for physiology, right? Because I remember doing my physics labs, like looking at little cars go downhill and measuring the speed, the velocity, and it was so boring for me. But when you think about respiratory, there's a lot of laws of physics that we can describe the processes with. So the one that I told you about the container is Boyle's law. And you're going to see that there's other laws in my chapter, in my narrated PowerPoint, that they have to do with physics. But in this particular one, it's just breathing, right? Taking air in and out. So when your volume increases, so it will be this one. This is what happens when you inhale, when the volume of the container increases, and you can say the container is your lungs in your thoracic cage, right? The pressure inside decreases. So why? Because if you have the same amount or particles of air here, you can tell that if I made the container larger, the particles are not that close. And if they're not that close, they're not exerting any pressure against them. So that's what lowers the pressure. So it's inversely proportional, right? The volume increases, the pressure decreases, because now the particles are spread out. And that's what we do with our inhalation. Now, when you exhale, we are on this other end. When you exhale, and if you exhale and put your hands on your rib cage, your ribs go in, and your diaphragm will become like a dome. So your whole container, lungs and thoracic cage, decreases in size. So the pressure is higher because now the particles are closer, so they're exerting more pressure. So now the pressure, the air pressure is higher on the inside than on the outside. So when we are exhaling, we have about 762 inside. Outside, we still have 760. So now we're going from high on the inside to low on the outside. So that's why I said that breathing, taking air in and out, is a very physical process. It's just the laws of physics and mostly explained by Boyle's law. Boyle's law says that the higher the volume, the lower the pressure. The lower the volume, the higher the pressure. That's what the law says. And that explains what happens in our lungs with our respiratory muscles increasing or decreasing the size of our container. Is that good? Yeah. Okay, so here you have the different pressures, and this is your normal breathing. And like I told you, you have, when you inhale, how the diaphragm is more flatter, and the ribs are pulling your rib cage up and out. So that's what makes it larger on the inside and lower for the pressure. So 758 and 760 outside. So that's how the air goes from high to low. Very easy. So what you're going to do in this lab, you're going to simulate what the volume of your lungs and pressure changes occur. You're going to be using a very old model, which is called the bell jar model, that it is to represent your lungs. So the lungs are going to be balloons. The diaphragm is a rubber sheet. Actually, the diaphragm is a sheet muscle. And the thoracic cage is going to be like a glass bell over it. And the trachea and the primary bronchi are going to be like a glass tube in the form of a Y. So this is what you're going to see in your little virtual simulation. So these are your lungs. These are your trachea and bronchi. The bell is the thoracic cage, and the rubber down here is the diaphragm. So you're going to be increasing and decreasing, or doing the inhalation, exhalation, and you're going to see the little balloons inflate and deflate. So that's what you're going to see in that simulation. Kind of like representing Boyle's law, just interpreting how the air goes in and out. The next one you're going to do for respiratory is called lung volumes. So lung volumes is a series of lung air amounts that the respiratory therapist can measure on a patient, and it can determine or tell the physician if the patient is suffering from any lung disease. And there are two kinds of lung diseases that are kind of like overall two major ones. One is called obstructive and the other is restrictive. Okay, so both are not good, and both are going to give the doctor understanding the different volumes. Both are going to give the doctor a good idea what's going on. And let me close my door because my dog is going nuts. We're still going to hear her, but less, little muffled. So what we use for this is called a spirometer. And if you look at the one that is in the virtual, this is a handheld spirometer. It's just on this area over here, the patient is gonna blow, and you're gonna see the volumes right here on the screen. When you do it with a physical therapist, it's a larger machine. They plug your nose, and all you have is the mouthpiece, and it's a little more involved type of equipment, but they're measuring the same type of lung volumes. And so that's what you're gonna do. You're gonna do spirometry on several patients, and you're gonna see the pulmonary function of these patients, and maybe diagnose what pulmonary diseases the patient has. So you're going to have a chart for the lung function, what each of the lung volumes is reported, and then you're going to have a chart for what kind of disease the patients are indicating according to the different symptoms. So when you have what is called an obstructive, I'm just going to move this. Oops, sorry. Did it wrong. So an obstructive disorder is where the flow of air coming in is less than the normal, or should be. A restrictive disorder is normal flow, but with a low vital capacity. So we're going to see what vital capacity is, which is the overall function of the lung. And it also has a low inspiratory and expiratory volumes. So we're going to go through each of the volumes that you're going to measure, and you're going to see the difference between the two conditions. So these are the different lung volumes that respiratory therapists will measure on a patient that is suspected of having either a restrictive or an obstructive respiratory disorder. The first one is called tidal volume, or TV. V is TV with V, right? Not B. This is the amount of air that is exchanged in a regular respiratory cycle. So it's an in and out exchange. And that's about 500 milliliters of oxygen. This is average, of course. Female sometimes is less, males is a little more, but this is during regular respiratory cycles, resting, all right? Inspiratory reserve volume, IRV, is the additional volume of air beyond tidal volume. So if you're breathing normally, and then you say, how much more can I breathe in? That's inspiratory reserve volume, okay? So this is the additional beyond the tidal volume. The expiratory reserve volume is the opposite. So when you're exhaling and then you can force more air out, is beyond what you will normally exhale in tidal volume. That's what is called expiratory reserve volume. And the vital capacity is the sum or the addition of all three. It's your tidal volume plus your inspiratory reserve volume plus your expiratory reserve volume. So I'm going to show you a graph because I think that a graph is, I'm very visual, so I like graphs. So here you have, at the bottom here, we don't have the legend, but that will be milliliters of oxygen. No, this will be time, sorry. This will be milliliters of oxygen. So tidal volume was the first one that we mentioned. Tidal volume, or TV, is the amount of air that we are exchanging in a normal breathing cycle. So you inhale, you exhale, inhale, exhale. So that's what tidal volume is. Inspiratory reserve volume is any air that we can inhale beyond the tidal inhalation. See, so here will be my tidal inhalation up to here, but if I force it, I can inhale more. So that's what we call inspiratory reserve volume. And same with expiratory reserve volume. When you're exhaling your tidal volume, whatever more air you can force out, that's considered your expiratory reserve volume. Now, when you add all three, your tidal volume, your inspiratory, and your expiratory reserve volumes, you add them all up, and that's what your vital capacity is. Vital means to sustain life, okay? And then we have always about 500 mLs of oxygen that is called residual volume. This air never leaves your lungs, and it cannot really be measured unless if you have a punctured lung, then your lung is going to collapse because you don't have any residual volume right there. But we know that we always have to have that little residual volume. So if you add vital capacity to the residual volume, then that gives you your total lung capacity. Yeah, so looking at this graph, I think it's a little easier to understand the different lung volumes that respiratory therapists will measure and send to your specialist to see how good your lungs are. Then there's other types of measurements that the respiratory therapist can do if he's asked to by the doctor. One is called forced vital capacity. So this is the total amount of air that is forcefully exhaled from fully inflated lungs. So it means that you took an inhalation, then you inhale a lot more, and then you forcefully exhale all that. So that's what is called forced vital capacity. So it's all the air that you inhale, tidal volume plus inspiratory reserve volume. You're going to exhale it. Again, tidal volume, expiratory reserve volume. So that's what we call forced vital capacity. Then we have forced expiratory volume 1, or FEV1. And this is the amount of that FVC in 1 second. So this is something that you have to, again, a spirometer, if you think about it, is measuring exhaled air, right? It has a direct relationship to how much air you inhale, but what you're really measuring is whatever you're exhaling out. So this is how much air can you exhale very fast in 1 second. And then there's another one that is not as common, which is the forced expiratory flow 2575, which is the average flow rate of the middle half of the FVC. So they really never do this one, but yes, they do the FEV1. They want to know how fast you can exhale all that air that you inhaled in. So this is the flow volume loop comparison to tell you how the obstructive or restrictive diseases are. So normal, you can see the normal flow of the air. And as I said, we're measuring exhalation, right? Because we have a mouthpiece. We can't really measure inhalation because you have to put a probe inside your lungs, and that's not very easy, right? But we're measuring exhalation, but it has to do with your inhalation, right? If you inhale more, you're going to exhale more. So this will be normal when you have this flow coming down like that is the normal flow of that exhalation. If you have an obstructive disease, look what happens. You have some pressure here and the flow is impaired. It's almost like breaking up. So you're like, cannot really exhale all at once. And then when you have restrictive, you can see how restrictive the space or the container is, right? So the flow is less It goes in a normal fashion, but it's a lot less coming out because we have less of a surface area to fill with air. So that's how you can determine an obstructive from a restrictive disease. So they do this loop analysis, the blood flow volume loop analysis, and they do the volume test and the FEV1 and the FEB, there's a typo there, FEB20x1080, and they can differentiate between the two diseases. All right, any questions on our little labs? They're fun. Just, okay. No, I'm not gonna share. So any questions on those that we've seen? Have you done it, some of you? Yeah, all right. If not, they're due tonight. You didn't know. And if you did the ELISA and forgot the worksheet, that I show you where it is, right? The worksheet. All right. So we still have some time. I wonder, do you have any questions on the exam? That's good. Chapter 12, 13, and 14, right? And then let's look at what are the labs that we're gonna have for our exam. Okay, so we did chapter 12. So we're gonna have skeletal muscle 1 and 2. Remember those? Bicep and then we're gonna have all the blood vessel, blood and cardiovascular. These are quite a few. So differential count, hematocrit, hemoglobin, blood typing, blood pressure, electrocardiogram, and pulse rate. And that's about it, because we're not doing chapter 15 or 16 for the exam. So that's about it for the labs. Should I just post this just in case? The labs are going to be involved. Some people are not here. I still have 30 people on my roster, but only 24 here. What else I was going to say? Again, the exam is going to open Tuesday at 8 a.m. and it's going to close on Wednesday at midnight. And remember, we don't meet on Tuesday because you're taking the exam. And what else? I think that's about it. Any questions on the exam? No? Study guide? Haven't done it yet. Of course. Why? It's a week away. Okay, so I'm not gonna do chapter 15 because I don't wanna get a lot of information in your head. I can go over that either during an office hour or next week because I really wanna, the following week, I really wanna go over respiratory with you guys so that we can see the relationships with cardiovascular, because once we have that, respiratory and urinary, you're gonna see the big picture, okay? Immune is great, it's nice, but I want you to really get those three, yeah, and understand the relationships with those three.